Welcome back to Dr. Liu's VCE Biology. Today's topic is the regulation of biochemical pathways from the VCE 2019 Biology exam. Let's learn together. If teachers, parents, or students see mistakes in my explanations, please give me your feedback. Protein activity may be affected by the temperature and pH of its environment because these factors change the protein's shape. The primary structure of a protein determines its three-dimensional shape, but the primary structure itself is not the three-dimensional shape. Proteins play a key role in the immune response. Some proteins with a quaternary structure are not enzymes. Acetylcholinesterase is an enzyme that catalyzes the breakdown of the neurotransmitter acetylcholine into acetate and choline. Aldecarb is a reversible inhibitor of acetylcholinesterase. Reversible inhibitors function by temporarily blocking the enzyme's active site, forming a weak bond rather than a permanent one. This reversible binding decreases the enzyme's reaction rate and reduces its overall activity by lessening the number of active sites available to substrates. Relenza works against the influenza virus by targeting a specific surface protein. This protein, an enzyme called neuraminidase, is essential for ongoing virus replication and is resistant to change. Mutations in the DNA of some resistant influenza viruses caused changes in the shape and charge of the enzyme required for viral replication. As Relenza inhibits replication by targeting this enzyme, these mutations can lead to drug resistance. The experiment investigates how temperature affects lipase activity, critical for biological and industrial processes. Lipase breaks down fats into glycerol and fatty acids, neutralizing alkaline solutions like sodium carbonate. The setup involves 4 test tubes with fatty solution and sodium carbonate at different temperatures. After adding lipase, pH is monitored from 8 to 7 using probes. The procedure is repeated 3 times for accurate and reliable data collection. To identify the most precise temperature from the data table, calculate the difference between the maximum and minimum values for each temperature tested. The temperature with the smallest difference, least variation, indicates the highest precision. Based on the data, the least difference was found at 35 degrees Celsius, making it the most precise temperature. The difference between quantitative and qualitative data is that quantitative data is expressed numerically, while qualitative data is descriptive. Directly write the conception and don't use an example. No mark was awarded for quantitative without explanation. Errors are present in the measurements taken at 15 degrees Celsius and 25 degrees Celsius. These errors are identified because the range of data, difference between maximum and minimum values, for these measurements exceeded the acceptable limit. Potential sources for these errors include adding an incorrect volume of lipase or sodium carbonate, incorrectly measuring the volume of fatty acid solution, inaccurate measurement of pH, or incorrectly recording the time. A systematic error is evident in Student 3's results, as all recorded values are consistently lower than the previous data. Potential causes for this error could be an incorrectly calibrated pH meter, or the consistent incorrect measurement of the lipase solution volume using a measuring cylinder. The limitations of this experiment include the restricted temperature range studied, as measurements were only taken up to 35 degrees Celsius. A wider range of temperatures should be investigated in future studies. Another limitation is the absence of a control. A sample without lipase should have been used to provide a baseline for comparison. Accounting for 15 of the total 120 assessment marks, 12.5% of the assessment, the Regulation of Biochemical Pathways section consists of 4 multiple-choice questions worth 4 marks and 1 short-answer question worth 11 marks. If you need my help, you can add me on WeChat or WhatsApp, or send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.